Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today we will talk about Boltz-1, or in fact I will teach you how to use Boltz-1 for your structure prediction. What actually Boltz-1 or Boltz is. So we will discuss all these things in the next slide. Okay. Boltz, or you can say Boltz-1. This name might be a new for you, but let me tell you, it's a new tool in the research arena, and it uses the power of machine learning and artificial intelligence. It gives performance and accuracy almost equal to that of AlphaFold 3. Above all, it is developed by an research group working in MIT, and it is an open source. It means it is freely available. And most importantly, their paper is still under consideration for different journals, and it is available in the preprint. I will share in next slide. That came to the BioRex IV. It's a preprint server, and on November 20th. So boards released this month, and I'm trying to teach you how you will use boards if you are unable to use AlphaFold 3. But before that, I want to emphasize the importance of open source, like what actually open source is and what is its importance. AlphaFold came in 2018, but they did not release their code for version 1. People waited for 2 years until COVID pandemic came, and in the early months of 2021, when it was pandemic around the globe, so AlphaFold 2 came, and by that time they released the code publicly. Thanks to them, because it helped a lot of researchers across the world. Then last year they have released, I think at the start of 2024, they released AlphaFold 3, but they did not release the code. And I have already covered a video. We were only allowed to use that server thing, and now almost 10 days ago they have also released their code. So if a program or a suite is not open source, then at times you have to wait, or the developers are not allowed. From the developer, I means other people, the third party peoples like me, like you. If you want to include something more, and or if you want to use that program and to make it more powerful, you cannot, because that is not an open source. So with this regard, Boltz is an open source, and it gives you the accuracy and performance up to AlphaFold 3 level. This is their BioRxiv paper preprint posted on November 2024, democratizing biomolecular interaction modeling by this group, and it's the program name is Boltz-1. The one actually belongs to the version. You will heard this news so quickly, like Boltz 2, 3, 4, 5. So these will be the version. Like the next version of AlphaFold will be AlphaFold 4. So currently we are using AlphaFold 3. So if you are interested to learn about boards, how it is working, how much it is different in methodology and an algorithm from AlphaFold, you can read this paper here. You can find it on Google. So now we are coming for the actual tutorial of Boltz. So stay with me. Okay, this is famous protein sequence database, UniProt. I will pick a sequence from the UniProt and will add some ions and will add some ligands just randomly, just to tell you how things are working or how can you use bolts for your research. So as usual, I'm picking indium one of my favorite proteins. So from here, I will just pick randomly, let's say from Klebsiella pneumoniae, this one. Then I will click here sequence. In sequence part, I will need this whole sequence. And if you are not interested in whole sequence in your protein, then you have to copy this to the text file and then uh, edit that sequence. Okay, I also want to answer one question here. Most of the students are messaging me, or sometimes they ask me that in their protein they have a mutation. Let's say they have a mutation I to L, isoleucine to leucine. So they want me to tell them like how can you introduce such kind of mutation. So my dear, that is so simple. Copy this sequence and just open a text file. Let's say this one. Let me clear. Okay, this is my sequence. I copy from here and with Control V or right click paste, you can paste it here. And then let's say this I is L, so make it L. That's it. And then copy the whole sequence and paste it into your favorite program. Okay, but here I'm going to take this sequence as it is. Okay, how I forgot to open the board's place, how you will run it. Okay, we will run boards in the Google Colab. So how can we run it? Just click here, type here col colab.research.google.com/, github /ash 100. It will lead you to my repository. Now here you will see like this ash100 /ai me. Okay, so if your internet connection is not powerful and you cannot see it clearly, no problem. I will drop this link into the description section of this video. Okay, from here, you click here and select DAS ash 100 /DAS. Once you click here, you will see this thing, Boltz-1 dash structure prediction tool. Click here. Now you are here. Believe me, sorry. Remember, this is a kind of alpha version of this notebook. From the alpha version, I mean that you will experience some new cells here and some new addition in coming future days. Then this notebook will be enhanced further. Okay, so you will like it. So far I will present you that how you can use this sports program. Okay, click here, connect. Try to connect it with from the GPU. From here, runtime, change runtime type. If you are a free user, your option will be here CPU. I would suggest to click here T4 GPU. Okay, so that's better. Now you can see query sequence. So if you are new to this video, I would suggest to please concentrate for some explanation. Okay, what are those explanation? Okay, I'm using this sequence. Let's say copy it. Just paste your sequence here. Okay. Now, if you are handling more than one protein. What will you do? Because you cannot see another line for a query sequence, then you have to just enter colon, not semicolon, colon, and now paste your another sequence without any gaps. Okay, point one. Point two. If you are working with a homodimer or heterodimer or whatever, so you will do the same thing. This colon will initiate a chain breaks. Chain breaks, I mean, for example, if you have two proteins or two domains, you want them into a separate chain A and B in your PDB structure, then introduce this colon between those two sequences, and the program will recognize by itself, or the program will take care of those sequences individually. So don't worry of that. Okay. This was point one. In my case, I'm going to model only one chain. Okay, this is ligand input. Okay, so what is your ligand? From the ligand input, that means anything which is not present, like your drug, your inhibitor, your metabolite, whatever you are trying to model. So in this case, I will use just PubChem. If you don't know, so PubChem is a database of different organic and synthetic compounds. We will pick any compound randomly. Let's say I will pick ibuprofen. So I will pick this compound. Here you will need to copy this isomeric smileys of that compound. Control C. And now paste it here. In case you have more than one compound, once again insert colon, then smileys of another compound. But I would suggest to test or to run this for the first time using one compound. Okay, here is ligand input CCD. From the CCD, actually simply that means that the ligands which you want to import from the PDB. Let's say this protein I'm dealing, it contains zinc ions. So I'm also interested to place zinc ions. So in PDB, the name of zinc ion is written this way. Okay. And how many zinc ion I'm interested in? Three. So sorry, two. So zinc and again Zn. It means it will place two ions. Okay. Ligand input common name. Okay. So it's up to you. Let's say to specify multi-ligand, the CCD code 3 letter. It's okay. Okay. If you are using this ligand, then enter a name here. So from where you will enter this name, for every compound there is a bigger name or also smaller name for some. So I will just copy this and place it here. Okay, now if you are dealing with DNA or RNA sequence, whatever, so drop your sequence here. But once again, DNA is a double stranded, so you have to place two times that specific sequence separated by this colon. But I don't have a DNA, so I will leave as it is. I will name this job, let's say, Bioinfo 1. Okay. So now click here. You will see this message. Don't worry. Okay. Now program has recorded my entry and it say I have this one ligand. I say, okay, now you need to click here, install dependency. It may take a minute or two depending upon your internet connection. So therefore, I will pause this video for a while. Okay, that's it. With me, it took almost more than a minute. Now we will allow the program to generate multiple sequence alignment. So now it's generating, and in the meanwhile, uh, I will announce something. In case you have some work and if you don't have enough expertise or if you don't have enough time, so you can handle that work to us, um, just like simulation, docking, and many other things. So we are working with a smaller company, kind of a company, research company. So if it is feasible for us, we will be able to help you out. Okay, then we will run this cell. Let's come back. It's done. You, by the way, you can clear this cell if you want. Click here and clear selected output. No problem. Okay, predict structure using bolts. Click here and we will wait for almost a minute or less than a minute. That's okay. And once it is done, we will also compare the structure of generated by boards with the PDB structure we have already 4EXS, but that is solved with captopril, L-captopril with the ligand here. And let's see how boards will place our zinc ions and will place that ibuprofen, where it will place. So we will all analyze this thing in this video. It's predicting. Okay, I'm going to pause it for a while. Okay, that's it. It took almost 2 minutes or 1.5 minutes. Okay, you can clear it. Now we will analyze our results, and before that, I will generate a directory on here and I will name it boards. Okay, whatever it is. Okay, and now I have also the 4ex PDB. I will bring it here to the bolts directory. It's the crystal structure. We will compare it shortly. Okay, now download results, run this cell. Result name is Bioinfo1 and result will be downloaded in zip form. It's here. I will cut it and will bring it to the desktop, this directory boards. Okay, paste. Now let me extract here. Okay, so I have already in the start I told you that this is a preliminary version because just a week ago your research paper came, so it will take few more days or few more weeks. So this notebook will be updated gradually and we will be able to predict more than one structure and to analyze that structure based on the PLDDT or some PAE matrices like how much error the structure has, blah blah. So right now these things are in pipeline. Okay, this is you will receive this by default this alignment image, like how much alignment, how many sequences were used, and in which area of sequence the alignments were weaker or stronger. It's okay. Okay, now I'm going to open my visualizer and I will keep this directory in path. So as a result, cd this directory. Now this is my wait, this is my visualizer. So let's open. So I have four EXS and one CIF, only one model, the best model it gave. Right now, it did not provide any opportunities like how many models we can predict, but soon we will have this opportunity. Okay, so first of all, I'm going to open this boards model. So you can see, let me make the background white. Okay, now you can see two zinc ions in the active pocket and two ibuprofen molecules were docked. Why two? Because we have entered only one ligand, but it has docked two. Because boards believe both of these can be correct poses. So and it is high likely because in natural system proteins are not static and they moves from here and there. So according to boards, this one is also the correct confirmation. Let me color it differently. Sequence, then two ligand. uh, I will color one red for better visualization and one yellow maybe. Okay. Oh, sorry. Okay, both are yellow, it doesn't matter. Okay. So you can see this is one ibuprofen, this is another one. So you have to remove one or you can keep both. It's up to you. Now I'm going to load the PDB structure. Open 4ex and it's here. Okay. This is the PDB structure, the actual PDB, and L-captopril is here with two zinc ions. Now I'm going to align both of them to check how much perfect we got the result. Okay, align it to 4x. Now you can see the green one is from predicted by the boards, the cyan one is predicted by the uh, X-ray crystallography. Now you can see the position of two zinc ions perfectly fine to me. You can see this, let me zoom it. You can see the cyan ligand that is perfectly dipped down between the two zinc ions. So one of our ligand is here. Exactly the same conformation as captured in 4EXS of captopril. And another one is here. So that is not an issue. We cannot say this is wrong because NDM1 has more than 40-50 structures in the PDB with different ligands like penicillin and other those cephalosporin beta-lactam antibiotics. So if we analyze with or if we compare it with some another one, possibly you will see that no, this one is fine. So the good news is that both of the ligands are docked inside the active pocket correctly. Also, both of the ions are docked perfectly fine to me. And overall, the structure match are aligned perfectly fine. That is amazing. So that was it. Like from now on, you will be able to use boards for your molecular structure predictions or for molecular structure. I will also say docking. Okay, and I hope this you will find this video a good one. If you like my work, if you're liking my work, then please subscribe to this channel and share this channel to your other colleagues. If you have any question, please drop your question in the comment section. For sure, I will answer. Okay, see you. Bye bye.